Hello, good afternoon, everyone. Hope you had a good weekend. We are going to be finishing up lecture 5 today. So last time, we did a summary of, the process of cellular respiration, and then we went into details of the first three stages, which included glycolysis, followed by pyruvate oxidation, and then there is also the Krebs cycle. Okay? And what's important to note is that at each of those stages, we actually produce some ATP, not a lot, just 4 net ATP in total, and we were continuously removing high energy electrons from the glucose. And all these electrons are going to be captured by energy carriers such as NADH and FADH2. And we're going to be taking them to the ETC to produce energy. So let's go to our study guide here for a second, and let's write down the key ingredients for each step as a summary, and then I'm going to tell you what happens at the electron transport chain. Okay, if you don't have the study guide, that's fine. Just take out any piece of paper and copy what I'm about to write. Okay, so stages in cellular respiration. There are 4 main stages. First one is glycolysis. Glycolysis. There we go. Um, as you might recall, glyco means glucose and lysis means to break. So glycolysis is going to be breaking down glucose into 2 molecules of pyruvate. Where does it take place? Okay. This takes place in the cytoplasm of the cell. Okay, and this is considered as anaerobic reaction as it does not require oxygen. Some of the key products we have include 2 net ATP. Okay, we actually invested 2 ATPs to energize the glucose, and in return we got 4 back. So 4 minus 2 gives you 2 net ATP. In addition, we are going to generate some NADH, 2 of them, okay, and 2 pyruvate. Okay, pyruvate has 3 carbons each. So I want to put a star here and remind you that these ATPs are produced by substrate level phosphorylation. That is the method where an enzyme comes along and it will transfer a phosphate from a donor, from a fuel molecule, onto ADP. And in the process, you produce ATP. This is not the most common way of making ATP. Only 11% of the ATPs are produced this way. So that's glycolysis. Stage 2. Okay, stage 2 is going to happen if there are sufficient oxygen available. And this is called pyruvate oxidation. And you will oxidize pyruvate. You will remove more electrons from the molecule. Does anybody remember where does this take place in the cell? What location does pyruvate oxidation happen in? If you have the notes from last class, that might be a good place to take a look. That's right. It is going to happen in the matrix of the mitochondria. Very good. All right. And pyruvate oxidation is going to produce 2 carbon dioxide, 2 NADH, and at the end you will get to acetyl-CoA. Okay, there are three things that happens, right? Each pyruvate will um, lose a carbon dioxide. You remove high energy electrons from it, and you will capture them with NAD plus, creating NADH. And then you will add an energizing molecule called coenzyme A, creating acetyl-CoA. Now, I just want to put in bracket here, all these products, it's per glucose, per glucose. We also did a summary chart last time, I think. It's probably like this one, right? Okay. Where we wrote down all the steps that are happening. Okay. Maybe I forgot which one was yours. Okay, but like something like that. Okay, so like it tells you what you're creating in each case. Okay. Uh, Next we have the Krebs cycle. Krebs cycle, also known as the citric acid cycle. And it also happens in the matrix. The key products per glucose include 4 carbon dioxide, 6 NADH, 3 per spin, 2 FADH2, and 2 ATP. And again, I am going to put the same purple star here because these ATPs are also produced by substrate level phosphorylation, where an enzyme comes along, transfer the phosphate from a donor or fuel molecule onto ADP. Okay. Only 4 ATPs are produced this way, 4 out of the 36, which is about 11%. And that's sort of where we ended the story. Okay? Now, what's important to kind of note is that all these energy carriers, they are going to transport the electrons to the ETC, to the last step. Okay? Electron transport chain. Electron transport chain. And it happens in the cristae, which is like the foldings that are formed by the inner mitochondrial membrane. And the key products that we're going to get, like we haven't talked about it yet. I'm just going to write it down for you and then we will, explain. I'll explain it to you. You're going to produce 6O2, okay? And you're going to produce 32 ATPs, okay? So these energy carrier will go to the ETC, will drop off the electron, and then they will get turned back into the empty version, right? The NAD plus and then the FAD so that they could go back to the first 3 stages and pick up more electrons. And remember, these energy electron carriers, they are like taxi. They go back and forth to pick up the electrons and drop them off at the ETC. So that is a quick summary. And just to wrap this up, these ATPs, they are produced, I'll put it here, produced by the second method. Okay, we wrote it down last time. It's called oxidative phosphorylation. Okay, I did not explain to you what it actually is. I just told you that this is the main way of producing a majority of the ATP. Right? I think at the time I told you it's going to take like half an hour to explain, right? And that's what we're going to do right now. Okay? But here is the summary, essentially, of everything that we've learned. last class. Does anybody have any question before we proceed? All right, so let's start a new note. Today is October 7th. You guys are 201. Okay, so here we go. This is gonna be a big drawing. If you have a piece of paper, then I want you to draw it like this, okay? Draw it like that. And then we're gonna maybe write some note on the side here. Okay? So let's do it. So here we have a line, and then I'm gonna do it like a wavy thing here. Right? So out here, this is the cytoplasm. Please stop me if I'm not making sense, okay? This part is like difficult to understand. Um, but try to draw it out with me, and if you have questions, do stop me. Okay? And this here, in case you're wondering, that's the outer mitochondrial membrane. This here is the inner membrane, which forms the cristae, the foldings. Okay. So what do we call the space here between the outer and the inner membrane? Does anybody remember? Like I know it was like a long time ago, but just try my luck here. What do we call this space? Thank you, Elvis. That's very helpful. This is the intermembrane space. And of course, this would be the matrix. Okay. Now, don't draw this next part, but basically, this is the mitochondria, and then we are zooming in. And we're zooming in to see, what's going on in there. Okay. Let's give you a moment to catch up. And yeah, we'll continue when you're ready. All right. So next we're gonna draw some proteins here. Okay. We're gonna draw three like that. Okay. And then we can maybe draw another three here. Okay. So there are like thousands of these things throughout the entire cristae. Remember, the cristae folds up. It's gonna be like the entire inner mitochondrial membrane, right? And then you have these proteins that are embedded within the inner membrane. So this here is what we call an electron transport chain. Okay, it is basically a series, we can say a group, okay, a group of membrane protein. Each subsequent protein has a higher electronegativity, which means attraction for electron. Okay, now I'm just gonna, I mean, they have names, but we're not gonna worry about them. Okay, I'll call it like one, two, three. Just listen for a second, okay? And then you can copy. Number two is gonna have a stronger attraction for electron than number one. And number three is gonna have a stronger attraction for electron than number two. Okay. That's what electronegativity means. Like, how much does it love electron? If it loves electron a lot, then we say it has high electronegativity. If it doesn't really want it, then it says low electronegativity. So basically, two loves it more than one, and three loves it more than two. Right? So if you, this is the most important part, okay? Listen carefully. If you give an electron to number one, number two will take it from one because it likes it more, right? And then number three will take it from number two because it likes it even more. And so it forms a transport chain for electrons because essentially, when you drop off an electron, it just goes, it's like the game, right? Hot potato, right? You take it up and then you pass it to the next, and you pass it to the next, and you pass it to the next. So one picks up the electron, passes it to number two picks up the electron, passes it to number three, and so on and so forth, right? Does that make sense? I hope it does. All right. So we'll come back to more drawing later. But I mean, like, you don't have to tell me if you don't want to, but like in your mind, you should think about this. What is actually going to be depositing the electrons? What is actually going to be giving the electron to number one here? Like, where do we get the electrons from? If you know, you can tell me. If you don't want to tell me, you should at least think about it. Okay. Where do the electrons come from? Yeah, from the energy carrier, right? Remember all those electrons that we capture from step one, step two, step three? All these NADH and FADH2, well, mostly NADH, right? Not so much FADH, like two of them, right? But still, right? They are going to drive the electrons. They're going to bring the electrons all the way to the ETC and drop them off. Okay? And that's what's going to happen, right? We're going to have the NADH, NADH here. It's going to drop it off, drop the electrons off. And once the electron gets off, once the passenger gets off, the NADH become NAD plus once again, right? And then this will go get more electrons from one, two, or three, right? Glycolysis, Krebs cycle, and pyruvate oxidation, right? Get more, and then they'll bring it back. Is that okay? Like, do you guys understand the relationship of what's going on here with the first three steps? Again, you can ask me anything. I'll answer it if I know the answer. Okay, thank you. So the electron is gonna hop off onto the ETC. I'm just gonna do it here with the orange pen here. And like I told you before, because of how the electron transport chain is arranged, like the next protein will take the electron from the first protein. So electron is going to get passed to the next one, and then it's going to get passed to the next one until it gets to the end. Okay. Remember, these are high energy electrons. So as the electron is being passed down the ETC, the proteins, the orange ovals here, the proteins in the chain, is actually going to use the electron's energy to pump hydrogen ions from the matrix into the intermembrane space. Okay, so we have hydrogen here. We're going to pump it over there. There's going to be another hydrogen here. It's going to get pumped over here, and then hydrogen, and then it's going to get pumped over there. Okay, so I'm going to write it down over here. As electrons travel down, the ETC its energy are used to pump H plus from the matrix into the intermembrane space. So this is the matrix in here. This is the intermembrane space. And then we are pumping H plus over. Now, as we keep on doing this, over time, you will have high H plus here and low H plus here, right? And that's why this is an example of a active transport. You're trying to move things from low concentration to high concentration. It requires energy. So where are we getting the energy to pump these things? From the high energy electrons. Okay. So I'll give you a moment to think about that. I just remember you guys have a quiz for me today, right? That is for lecture 3 and 4. All right, so make sure you take some time to complete that before the end of today. All right, so we have a lot of H plus in the intermembrane space and lots and not as much in the matrix, right? So where do you think the H plus would tend to go? All right, here I'll put more H plus here, lots of H plus, right? Because we have been pumping them here into the intermembrane space. So things naturally wants to go from high to low, right? So all these H plus, they actually want to come back into the matrix, but they can't because H plus is charged, and the inner membrane, as you know, is made of the same material as the cell membrane. So it creates a barrier that prevents the H plus from going in. Okay, so let me just quickly write another note here for you. As H plus builds up in the intermembrane space, they want to move back into the matrix. But they are blocked by the inner membrane. Okay? Anything that's charged or big can go through membranes, right? Now, to help you understand this, this is like how dam works, right? Here, don't draw this. Just listen, okay? It's like this. It's like there is a waterfall or something, waterfall, and then you build a dam. Right? So what does the dam do? The dam is gonna block the water. Right? The water is gonna want to fall down, but you can't because the dam is in the way, right? So what people can do is they actually dig a alternative path, okay, and then they put like a turbine here. Again, you don't have to draw this. I'm just trying to help you understand, okay? So after you dam off the water, you let the water flow through this path, right? And as the water goes through the turbine, it generates electricity, right? Generate energy. This is basically what happens. So instead of building up water, we are building up lots of H plus here. The H plus wants to get to a low point back into the matrix. It can't because of the inner membrane, the dam. And so it's looking for an alternate path to flow through. Right? So that alternate path that we have is this special protein. I'm going to use a different color here to make it special. I don't know, like green or something, okay? We're going to have a special protein that looks like a doorknob. Okay, that's how all textbooks draw it. And this guy is called ATPase. ATPase. Okay, now sometimes it has a different name. I'm just gonna write it here as well. It's also called ATP synthase. Okay, because I did not write all the questions on the test, where we have like different people teaching the same class. Okay, so some of the other teachers might use a different name. Okay, but ATPase, ATP synthase mean the same thing. So this is basically the turbine. This is basically the alternate pathway. So all these H plus, they're gonna come back in. They're gonna go through the ATPase back into the matrix. That's the only path. Everything else is blocked off by the inner membrane. And as the ATPase, as the H plus pass through the ATPase, it's gonna turn the ATPase. And in doing so, you're gonna be generating ATP. I run out of colors here. Guys, I'm just gonna go like ADP, and then there's gonna be P, and then as the ATPase turns, that's gonna create ATP. So this whole process of producing ATP, that's oxidative phosphorylation. Okay. We had the uncommon substrate level phosphorylation that produces only 4 out of the 36. And then the second way is this method. This is called oxidative phosphorylation because the whole thing is powered by oxidative reaction, essentially. Okay, so let's quickly recap. We're going to have the energy electron carrier dropping off electrons at the ETC. As the electron pass down the ETC, the energy from the electrons are used to pump H plus into the intermembrane space. That creates a collection of H plus in the intermembrane space, just like you create a collection of water when you dam a waterfall. All these H plus wants to come back in, right? They can't because of the inner membrane. So the only way to come back through is through this special turbine, special protein called ATPase. As the H plus comes back in, it's going to turn the green protein, is going to turn the ATPase, which in turn creates ATP. Just like as water goes through a turbine, it turns the turbine, generating energy, generating electricity. I just have 2 more things to add to this note. But any questions at all about any of these up to this point? Okay, well, what about FADH2? I haven't included it yet, right? So I want to ask you this to jog your memory a little bit. Each NADH can produce how many ATP? We actually did a few questions last time regarding this. How many ATP can each NADH produce? 3 is correct, right? So in this picture, I showed that you pump 3 H plus over, and then as the H plus comes back in, you create 3 ATP, right? So FADH2 is going to come along, and then it's going to drop off the electron and it's going to become FAD. But because FADH2, the chemical energy is less than the NADH, that's why it's not going to be able to drop off the electron, as high of a point. Instead, it's going to drop off the electron at the second protein instead of the first protein. So, you can still pump H plus, and it's the same kind of H plus as before. I just used a different color to show you. But because they didn't drop the electron at the first protein, they drop it later on, you're not going to have as much chance to pump H plus. Right? So instead of pumping 3H plus, you only pump like 2H plus. And when these H plus comes back in, that's going to be able to generate like 2 ATP instead of 3 ATP. Okay. So of course, this is going to go back, go get more electron from Krebs cycle. Only Krebs cycles can generate FAD, H2, right? Maybe look at the notes from last time. All right. I'm going to tell you a quick story to help you sort of remember this or understand the next part. So when my kids were younger, they go to the park, and then every now and then they would run into this problem, okay? Like this is the slide, okay? And then, I always tell them, wait until the slide is clear before you go down, okay? But every now and then there's going to be a kid who slide down the slide, and then, they stay there at the bottom, and then, clogs up the whole slide. Yeah, because the next person can't go, right? And then my kid would look at me and is like, I want to go, but I can't, right? So usually what happens in these situations is that, the parent's gonna come, okay? Parent, okay? The parent's gonna come and it's gonna, take the kid away from the bottom so that the next person can slide down, right? Right? So remember this picture, or this story as I describe the next part to you. The electron in this case are the kids, and the ETC, the proteins, is the slide. So the electron is going to slide down the slide, okay, and then do all these fantastic things in the middle, transporting H plus. But when it gets to the bottom of the slide, something has to come and remove the electron. Okay. In fact, if we use proper, terminologies, protein number 3 has the highest attraction for electron, highest electronegativity in the whole chain. So we need something that has even higher electronegativity to pull the electron off the chain. If we don't pull the electron off the ETC, the next electron will not be able to go down, right? Okay. So if like what happens? Like if the next one cannot come down, then we don't have the energy to pump the H plus. If we don't pump the H plus, then we don't have the H plus coming back in to create ATP, right? And then we're not going to be able to make energy and we die. So what's going to be there to pull the electron off? Well, that's going to be the oxygen. Okay. Do we have any other like sharp color here? I guess not. I'll just use brown here. All right. So O2 is going to come pick up the electron. It's going to combine with some of these hydrogen floating around and is going to produce water. All right. So I'm going to write it over here. O2 is the final electron acceptor that will remove electrons from the ETC. And that's why we need to breathe. That's why we need to continuously have oxygen supplied to our cells so that we can make more energy to keep going. If we don't have oxygen, then the electrons will be stuck on the ETC. They have nowhere to go. It's like that kid stuck at the bottom of the slide. Next kid can go. Next electron can flow through the ETC. No electron flow, no H plus buildup. And no H plus buildup means no ATP. Everything that goes on in the body, right, needs some sort of energy, right? Without ATP, we die. So that's the story here. Okay? That is basically step four, stage four of cellular respiration. And that's how you make a majority of your ATP, 32 of them. Okay. Questions? All right, so that's a lot of stuff here. You probably have to go through it again on your own, read the PowerPoint slides, maybe watch the recordings again. Just make sure you have a good understanding of it before the test, which is next Wednesday. Okay, it's just to give you an idea of what's going to be happening for the next couple of classes. This Wednesday, we will have the review for the test. Next Monday is Thanksgiving, no class, and then next Wednesday will be test number one. Okay, I'll post the information for test one later, but test one is going to be everything we've done so far. 1, 2, 3, 4, 5, as well as the beetroot experiment. Okay. No class next Wednesday. You just have to do the test. Next week is not the reading week. The reading week is the week after. Okay. Um, just check the calendar if you are unsure. Okay. That's that. So let's go back to our study guide here. And we have this table. Right, so I want to show you where the ATPs all came from. Okay. So I'll give you a moment to get that. Oh yeah, so like Renita asked, I know you guys have a chemistry quiz today, right? Like I mean, usually it's like it's hard for everybody to coordinate with everybody to make sure that there are like no quizzes on the same day. But I think I forgot which one of you told me last week, that you do have a chemistry quiz today as well. Actually, thank you for reminding me. So what I did was I actually extended the quiz day. Okay, for today's quiz, like I extended to tomorrow, so you can either do it today or tomorrow. Okay. So hopefully that helps a little bit. But like I said, like it's hard to coordinate all the time because like you might have a math test or you might have like a computer test or something, right? Like it's yeah. Anyway, I do try to help you guys out whenever I can, and so hopefully it's gonna be better for you. All right, let's do this thing. Glycolysis. How many ATPs are produced through substrate level phosphorylation? Okay, by now, you should know there are two net ATP. Oh, it's not letting me write it. Let me try again. Two net ATP. Energy carrier produced. Right? If you can't follow me, like if you don't know where the number is coming from, you should take out the note from last class. Okay, we took a note last class. Here, right? Like this tells you exactly how many of each things are produced. And so that's where I'm getting the numbers, right? So how many energy carriers are produced from glycolysis, please let me know.
Two,
And what are they?
two NADP,
Two NADH, that's right. Two
NDH.
NAD, yeah, two NADH. So that's what we're gonna write down here. Two NADH. All right. Now these guys are a little bit problematic, okay? I'm just gonna make a note here for you. The NADH produced in the cytoplasm, which is where glycolysis takes place, right? Cannot enter the matrix, okay? Because they're too big. Okay? They cannot enter the matrix, because you gotta cross like two membranes to get to the matrix, right? The outer membrane and the inner membrane before you get to the matrix, and it's just like too big, can't do that. So what happens is that um, instead of trying to cross the two membranes on its own, this is what happens. All right, so outer membrane, inner membrane, we have the NADH here from glycolysis. So instead of crossing by itself, which it can't because it's too big, it's just going to pass the electron over. Okay? And on the other side of the border, if you would, there's going to be the FAD, and that's going to pick up the electron. Okay, so this is going to turn back to NAD plus. So you might ask, hey, how come like we don't have another NAD picking up the electron? Why do we have to pick it up with the FAD? Well, nothing is free, right? So to speak. Whenever you do a transaction, you are going to lose some energy. Right? So when you move the electron over to the membrane, some energy is lost. And so you are going to pick it up with a lower, a molecule that has lower energy, basically. Right? Remember, NADH can make 3 ATP, FADH2 can make 2 ATPs. So in this transaction, you essentially pay the equivalence of 1 ATP to move the electrons over. Okay, so over here, 2 NADH, but like I said, these are going to become 2 FADH2 instead because of the transaction, because of the passing on of the electrons. And so when it does get to that ETC, we're going to times 2, and that's going to create 4 ATP. So from glycolysis alone, we produce 2 ATP directly, and then we produce 4 ATP at the ETC, which means in total, there are 6 ATP. Now let's think about the pyruvate oxidation. Again, look at the summary that we wrote down. Right? I mean, if you didn't write it down, and download a copy or something. So pyruvate oxidation, how many ATPs do we produce directly through substrate level phosphorylation? Okay, let's take a closer look. This is pyruvate oxidation right here. It happens in the matrix. And we remove the carbon dioxide, we produce NADH. We add the coenzyme A, but as you can see, we did not,
No ATP.
not produce any ATP directly. Okay, so we're going to go back to here. We're going to say no ATP. Not directly anyways. But indirectly, through the ETC, we have 2 NADH. We capture the high energy electron, going to transport it to the ETC. Okay. We don't need to pass it on to FAD this time because we are already in the matrix, right? This is already in the matrix. And this is cytoplasm. And what do I multiply by this time?
3.
3. Thank you. 3 times 2 is 6. 6 plus 0, 6. All right. Krebs cycle. So look at the Krebs cycle here. We have 2 ATP by substrate level phosphorylation, directly producing 2 ATP, 6 NADH, and 2 FADH2. If you have trouble memorizing these numbers, definitely have a cheat sheet with you on the test. Okay, it's going to help you out a little bit. So this is 2 ATP, 6 NADH, 2FADH2. We're going to multiply this by 3 and multiply this by 2, which gives me 18 and 4, right? So 18 plus 4 plus 2, that's 24. 6 plus 6 plus 24, 36. So each glucose can produce up to a maximum of 36 ATP under ideal conditions. Okay, if everything's perfect, everything is like going the way it should, then you're gonna get 36 ATP. All right? Any questions? All right. We're gonna go ahead and finish the PowerPoint slides. The rest is pretty straightforward, I think. But let's do a poll here. Let's do a poll. Sorry, just fixing up the answers here. All right, got like about half the class answering, not bad. Um, so a lot of people actually chose A here, and then that actually is not correct because, first of all, oxygen becomes water by picking up electrons, right? And remember oil rig that I told you about? If you gain electron, is actually reduction. Reduction is gain, right? So if anything, we say oxygen is reduced into water, not oxidized into water. Okay, it's the final electron acceptor because it is more electronegative than the proteins in the ETC. Remember, 2 likes it more than 1 and 3 likes electron more than 2. And then it's stuck at number 3, right? The electron is stuck at number 3 until oxygen comes along to pull it off. Well, how can electron pull it off number 3? It has to like electrons even more than number 3, right? It has to have a higher electronegativity compared to number 3. So that's the answer. Okay, here's another one. Should be pretty straightforward if you filled in the chart with me just now. Okay, never mind E. E is not the answer. We haven't learned that yet. Just ignore that for now. Okay, so glycolysis definitely the answer, and Krebs cycle, right? Those are the only places where we will be producing ATP directly, where we'll be, using an enzyme, ripping the phosphate off the donor and transferring it onto ADP. So only glycolysis and Krebs cycle. In fact, we wrote it down here, right? Only 2 purple stars, and those are produced by substrate level phosphorylation. All right, next we're gonna talk about, briefly anyways, the disruption of oxidative phosphorylation. In other words, when we disrupt the electron transport chain, what's going to happen? Well, the end results are the same. End result is that we don't make ATP, right? Or we don't make as much ATP, and that could be lethal, right? Because without ATP, your heart's not going to beat. Beating of the muscle that requires ATP. So, we die from that. But there are different ways, different kinds of poisons that can obstruct the ETC, interfere with oxidative phosphorylation. Just to reiterate, in case you're confused, okay? The ETC is, the proteins, the three purple proteins that we drew, okay? The method of producing ATP at the ETC is called oxidative phosphorylation. So, for us, they are kind of the same thing. If you block the ETC, then you basically block oxidative phosphorylation. Because without the ETC, you can't make the ATP, right? So there are a couple ways that can happen. Okay. Number one, you directly block the ETC with these kind of poisons. I'll tell you how. Maybe you inhibit the ATP synthase. And then the third way is like making the inner membrane leaky. So I'll talk about each one of these in details. Number one, all these things will block the ETC. Right? Rotenone, cyanide, carbon monoxide. This is carbon monoxide, okay? So rotenone is actually used as an insecticide and pesticides. They killed off insects and pesticides in places that you don't want them to be. And how it works is it binds to the first protein in the ETC, right? So in our drawing, we had like three purple proteins, right? Remember, I just called them one, two, and three. The NADH is supposed to drop it off at number one, and the electron's gonna flow through, right? So what this rotenone does is it will go to number one, bind to it, and blocks it. Then the NADH cannot drop off the electron, and you can't make ATP. Sure, the FADH2 can drop it off at number two over here, but that's like not a lot of ATPs at all, right? If you go back to the table we filled in over here in the study guide, look, only 4 ATPs are produced through FADH2. I guess like if you count these one as well, that's eight, right? That's nothing. That's like a drop in a bucket, right? Like it's not gonna be enough to sustain the energy need in the body. So that's why this is gonna be a poison. We don't want to get that into our body. Number two, carbon monoxide. Carbon monoxide is like tasteless, colorless, odorless. You can't smell it. You can't detect it with our senses. Okay, that's why we need to have a carbon monoxide detector at home, and you should put it where you sleep, right? Because that's when it's most likely going to kill you. And how it kills you is that the carbon monoxide binds to hemoglobin, which is the oxygen transporting protein in our blood. Right? So hemoglobin normally will deliver oxygen throughout your body, but in the presence of carbon monoxide, it will always choose carbon monoxide over oxygen. It's like my kids. If I ask them, "Hey, do you want to have salad or chicken nuggets for dinner?" 100% of the time they would choose the chicken nugget, right? Chicken nugget is the carbon monoxide in this case. Okay, when given a choice, hemoglobin will choose the carbon monoxide over oxygen. And of course, we cannot use carbon monoxide in the body. It's not going to be acting as a final electron acceptor. So the electron is going to be stuck on the chain. Next electron can't go. No hydrogen, no ATP. Dead. Next, we have cyanide. Cyanide is CN. You might know that from chemistry. And the poisonous form is usually hydrogen cyanide, HCN. Okay? It binds the protein in the ETC and basically inhibits it. Right? So same thing as the rotenone, okay? But the cyanide works slightly differently than the rotenone. We're not going to go into details. So all these things are poisonous. Okay. I don't have a slide on the oligomycin, but it's like antibiotics again. Like if people hear antibiotics, think of the drugs we take, but like not necessarily, right? Like antibiotics are basically any substance that are produced by anything really that can kill other living things. That's what an antibiotic is, okay? So in this case, it inhibits the ATP synthase. It inhibits the doorknob, basically. Okay, it blocks the turbine, and if you block the turbine, nothing flows through, then you don't make ATP, right? That's pretty obvious. The last one is the DNP. Okay? DNP stands for like 2,4-dinitrophenol. Doesn't matter what it stands for, but that's the name, okay? 2,4-dinitrophenol, DNP. It was actually discovered. Well, okay, so like backtrack a little bit. It was actually used as like a chemical for like fertilizers and like dyes and like explosives in some cases. Um, so the story goes like this, and you can look it up online if you're interested. That there was a worker in a factory that processes things like DNP, and he accidentally ingested some. Okay, accidentally consumed some of these DNP. All right, and what happens is he experienced very rapid weight loss in the next couple of days and weeks to follow. Okay. And so, when the doctor and the people find out about this, they thought they discovered a miracle drug for weight loss. Okay. And actually, it was marketed as by a pharmaceutical company as a weight loss compound for some time until they discover that it actually can be lethal if you consume like too much. And even a slight small amount, it could be lethal depending on the person. And what the problem with the DNP is that was that they cause the inner membrane to be leaky to the H plus. Do I have a picture for that? Oh no, I don't. Okay, but basically it's like this. Right? This is the inner membrane. Remember, you have all these H plus, and the whole point of having all these H plus is so that they're gonna go through the turbine, right? They're gonna go through the ATPase to create ATP, right? That's the whole point. With the DNP, if you consume the DNP, it basically makes the inner membrane leaky. Right? So now that it's leaky, the H plus is gonna go through, right? Like you have no dam. It's like a dam with holes in it. All the water is gonna leak through, right? So all the H plus is gonna leak back into the matrix without passing through the turbine, without passing through the ATPA. And so you produce much less ATP. Okay? With drop in ATP production, your body thinks you're starving. It's like, oh, maybe the guy's not taking in enough glucose. He's starving. So let's burn some of the fat for backup. Okay, and that's why there is that rapid weight loss effect. But of course, if you don't produce enough ATP, like weight is not necessarily the problem here, right? The problem is like your heart's not going to get enough ATP, your brain's not going to get enough ATP, and then you die. Okay, and people have died from consuming DNP. All right, so this summarizes what's going on. We have the rotenone, the cyanide, and the carbon monoxide all interfering directly with the ETC. We have the DNP that's helping the H plus come back in, bypassing the ATPase. And we have the oligomycin that would block the ATPase. There was a question in the chat. So why create DNP? Yeah, so like it was not created for like weight loss, right? It's created for like what did I say at the beginning? Like for fertilizers, for dyes and explosive. I don't know what it is that they use it for in those processes, but that was the intended purpose of these things, right? Of the DNP. Right? It was just like accidentally they found out, oh, weight loss, you will lose weight if you consume some of it. Okay. All right, we count the ATPs already, and we just have a couple more slides. So I think I'm just gonna skip the break and finish the slides with you, and then you can go on and do your chemistry quiz and whatnot. So cellular respiration prefers to use glucose. Okay. However, it's pretty versatile in terms of what it can actually accept as fuel. It can use different kinds of carbohydrates. Right? So glucose is not, it's a preferred kind, but you can also use things like fructose. Yeah, you can use things like galactose, and you can break them down and feed them into the pathway at different locations. Fat can also be broken down into the glycerol and then the fatty acids. The fatty acids can actually be chopped into uh, two carbon fragments, and they become acetyl-CoA. You don't have to memorize any of these. My point is that different fuel molecules, different macromolecules that we eat, they can be converted into different intermediate molecules that we can then feed into the cellular respiration process at different stages. Okay, so proteins, it's made up of like 20 amino acids. Depending on which amino acid we have, you can feed it into the Krebs cycle. You can feed it into the pyruvate oxidation and so on and so forth, right? So while glucose is the primary preference choice, right, for cellular respiration, we can burn a lot of these other things as well. And this picture basically shows you what I was telling you. The fats can be broken down into glycerol and fatty acid, and the fatty acid can actually be turned into acetyl-CoA. The amino acid, you can feed it into different stages depending on which amino acid it is. But ultimately, you're going to produce ATP. Like obviously, like depends on where you feed the molecules into, the number of ATPs you produce going to be different. If you feed the molecule into the Krebs cycle, you're going to miss out on ATP production from the earlier stages, right? So obviously, the earlier you are able to feed into the cellular respiration, the more ATP you can extract from the molecule. But generally speaking, like if you are having regular meals, uh, on a regular basis, ATP production tends to be not to be a problem. Okay. All right. There is a difficult question here. Yeah, I'll just go through it with you. I'll read it to you first, and then I'll tell you the answer. Hydrogen cyanide is poisonous. We already talked about this like 2 slides ago, but it binds to one of the membrane proteins in the ETC and blocks the movement of electrons. If cyanide poisoning occurs, what would happen to the pH of the intermembrane space? Okay, so let's recap what happens normally. Normally is you have the proteins and then you pump the H plus over, right? That's the story I've been telling you for the past hour. If we have hydrogen cyanide, we are going to basically interfere with this process, okay? And in doing so, we lower the amount of H plus. So with less H plus, it means that the pH is going to be higher. Okay, so there is a hint here. Low pH is caused by high H plus. So high pH is caused by low H plus. So the answer is this. Okay. Last, we are going to talk about anaerobic respiration. I'm just going to keep it simple here. The word anaerobic means without oxygen. Okay. There are different kinds of fermentation, depending on what organism we're looking at. Fermentation is useful for humans because, alcoholic beverages are made through fermentation, right? Like wines and beer, they are fermentation process. And if you use yeast to bake, the fermentation process is what causes the bread to leaven, to rise. So there are two kinds of fermentation. The type that happens in humans are called lactic acid fermentation. Lactic acid fermentation. All right. So This happens when oxygen demand is greater than oxygen supply. Right? If you're running at full speed after a minute or 2 minutes, right, 3 minutes, then you no longer can sustain that speed because your leg muscles are demanding more oxygen than you can supply. So without adequate oxygen, the pyruvate cannot move on to step 2, 3, and 4, right? Because the ETC is blocked, right? Without enough oxygen. So then your body, your cell switches to fermentation. The whole point of fermentation is to provide an alternate destination for NADH. Okay? You can't drop them off at the ETC because it's clogged up without the oxygen. So let's give it some other place to drop it off. Okay? Now, without going into too much details, basically the NADH that we create from glycolysis, now the pyruvate will use the NADH and create lactic acid. Now, this process is not producing ATP. Fermentation does not produce ATP. But what it does achieve is by freeing up the NAD plus. NAD plus can go back and allow glycolysis to keep on going. Right? So glycolysis, as you know, it doesn't generate a lot of ATP. It generates like 2 net, but it's better than nothing. It's gonna keep you going for like a little longer before you have no choice but to stop. Okay. Like practically speaking, let's say you're running full speed for 3 minutes and you're about to tap out. This process, this fermentation may allow to go on for extra, like, I don't know, 10, 15 seconds. Okay. So not a lot, but it's just something that your body does, right? Okay. It's like, doing curls. You already did like, 10 reps and this is your final rep, and then you're pushing through, and then maybe this process is going to allow you to finish your rep. Okay. So it creates lactic acid. And the buildup of these lactic acid, people say it's what caused you to feel the burn on the next day after you have strenuous exercise. Okay, so this is a picture that summarizes what's happening. For other organisms like yeast, which are quite simple, and for things like bacteria in some cases, they don't have the organelles and the structure to do step 2, 3, and 4. So they solely just do glycolysis. And it's okay for them because they're so small, right? They don't need as much ATP as us. And for them, let me show you the pathway here. Um, the glycolysis is the same as us, but instead of producing instead of producing lactic acid as a final product, they produce ethanol, which is drinking alcohol. And then that's what people want as a result when they use fermentation to produce alcohol. And at the same time, they produce carbon dioxide, okay, which is what caused the bread to leaven. Okay. So these are just alternate pathways for energy production. So basically, uh, the fates of pyruvate after glycolysis. Okay, there are three things to do. If oxygen is available, this is aerobic. Okay, aerobic means O2 available. Then we're going to go through step 2, step 3, and then produce 36 ATP. Fantastic. If oxygen is not available, or if the machinery, the tools needed to do ETC and all those things are not available, then the body, for us, we do lactic acid fermentation. But for the yeast, they would do alcoholic fermentation. We can't do alcoholic fermentation. Like imagine, like if we do alcoholic fermentation, we would have like, constantly have alcohol in our system, right? That's toxic to our cells and we're walking around drunk all day, right? So we do lactic acid fermentation. I there's a thing in the chat. I want to know if you can give a name of book. Yeah, so like again, people ask me this every now and then. But on the e-centennial, there is a link to the textbook, right? And let me just show you, um, for those of you who didn't know. So go to content and then lecture five. This is lecture five. And then there's the notes, and then there's the resource, and there's the supplementary reading. And so if you click on it, it will take you to the online textbook. Okay, if you don't want an online textbook, like any introductory biology textbook would have this stuff in it. Okay, so like you can just get one from the library or something like that. Right? So this is oxidative phosphorylation that we learned today, right? So that's the ETC. Now, the thing about reading the textbook is like I already make things a little bit simpler for you guys, right? I drew three proteins, right, in the ETC, but actually there are five, right? So like sometimes if you read it, maybe it conflicts with some of the stuff that we talked about. So that's why I said like if you're confused, maybe just listen to me explain it again. Or if you have a specific question, feel free to email me. But anyways, if you need, you can read about it. Okay, so that's the glycolysis that we did last time, and you can see they really list the details, right? Like 10 steps. So if there are extra information in the readings that we didn't cover, you're not responsible for them for the test. All right, so a couple of things. Tests, a quiz for you guys today or tomorrow. Just do it. Doesn't matter which day. I will post a review for the test, which is next Wednesday, and we'll take up the review together this Wednesday. Okay, that's it for now. Thank you guys. Good luck with the quizzes, and I'll see you on Wednesday for the review. Bye for now.
Thank you. Bye.